Alright, so now to do some problems. It's always a good idea for you to do problems. Either do the problems first and then use the book to help you figure them out, or watch the PowerPoint or read the book and then try to use the problems to reinforce what you've already seen. It's up to you how to do it. Um, we just did a video on chapter 11, and these are the most important things that I think are involved. So, describe the composition and structure of a nucleosome. Uh, Consists of a histone octamer. Now, I'm not going to write in full sentences or anything. Plus 146 base pairs of DNA. Histone octamer has H2A, H2B, H3, and H4, and two of each. And the histone octamer so then consists of 4 proteins times 2. So there's another version of that behind there. And then there's 146 base pairs of DNA wrapped around it. There are these tails, I'll make the tails green, that come off these histones, and those are going to be important. They come off the ones in the back too, and those will be important later. Not the greatest drawing, but it gets to the basics of what is important about nucleosome structure. Describe in steps how the double helix of DNA, which is 2 nanometers in width, gives rise to chromosomes at 700 meter in width. Alright, so that's going from DNA. So we got our double helix. It's more and more abstract, which is 2 nanometers. And we're eventually getting to a chromosome that's 700 nanometers. And so they're talking about that one chromatid is 700 nanometers. So what happens between here and there? Well, the first thing is the DNA gets wrapped around the nucleosome. To form the 10 nanometer fiber. Then this gets looped into the 30 nanometer fiber. So it's 30 nanometers this way. And then this 30 nanometer fiber gets attached to a scaffold, and then there's further scrunching of the scaffold to get to that. That's essentially it. This is the main important thing, is how it's going back and forth between the 10 nanometer and the 30 nanometer. So I left a lot of space there for some reason, but that's okay. So what are epigenetic changes and how are they brought about? We didn't. I don't think I mentioned epigenetic too much in the last video. I showed what they were, but I'm not sure if I straight up called them epigenetic changes. So the idea of epigenetics means that you've got a DNA sequence. So gene A, same gene, and maybe this is in the liver, maybe this is in the kidney, and this gene is expressed and this gene is not. So we have the same DNA, but we have a different outcome. So here's our phenotype, is that you end up with a liver cell. And the phenotype here is you end up with a kidney cell. So we're starting out with, so genetics means we start out with different DNA sequences and we get a different phenotype. So if you've got big A, you get one phenotype. You got big A, little a, you might get a different phenotype. Little a, you get another phenotype. This is saying we've got the exact same genotype and we're getting a different phenotype. And that within an organism, that's how different tissues develop, and it's by different expression. So it's not the genes, it's not the DNA that's causing that difference. It's how that DNA is expressed that's causing the difference. And so epigenetic changes are if we the gene um, in one cell in the liver cell is going to be loosely condensed. The gene in the kidney cell is going to be tightly condensed. And the reason for that is going to be modifications, or one reason for that is modifications of the histones. And so this modification of the histones is additional information on top of the DNA sequence. So same DNA sequence, but now we've got different histone modification, and that leads to a different result. But it's not the DNA that's giving you the different result. It's this histone modification, because this histone modification. So the genetics means the DNA. What's the DNA doing? Epigenetics means what else is going on. What in addition to the DNA? What is so this is the DNA sequence, and this is in addition. So epi means to stand on top of, and so the this layer of information is standing on top of this layer of information and sort of changing how that information is expressed. This is just one example of epigenetic modifications that can change gene expression, and we'll talk about more later. Chapter 21 is all about epigenetics. Euchromatin and heterochromatin. Essentially, euchromatin is found in centromeres, or I'm sorry, heterochromatin is found in centromeres and telomeres. So that's a centromere, that's a telomere. And if we can think about, so, and then this is euchromatin in the arms. And I don't know if I can remember everything that was in that little table, but get the general gist. The so location-wise, in the arms and centromere and telomeres. Now you can have heterochromatin dispersed throughout the arms as well, but we're just talking about the main sort of super obvious differences. Eukaryotic or euchromatic, euchromatin is where the genes are, and this is where transcription occurs. No transcription. This is decondensed or loosely condensed, and this is highly condensed. And they'll have different modifications. So maybe this is acetylated, and this is deacetylated. So that's the basic idea of what's the difference between these two things. There might be some other things. You can go back and look at your little table. Those are the main things. And this is the experiment that just looking at it, it's a little bit goofy. And but if we think through it, it's not too bad. So now we want to correlate changes in, I can't get this to be in the same spot. I apologize. Here, zoom out here. That might help a little bit. Right, so there's the question. Based on the DNA sensitivity to DNase 1 illustrated in figure 11.7, which type of chicken hemoglobin genes or chicken hemoglobin is likely produced in highest quantity at the following tissues and developmental stages? So all this is about, even though it might look a little bit complicated, the key is that these arrows stand for DNA hypersensitive sites. What that means is that there's loose chromatin, probably due to histone acetylation, although we haven't shown that, but just to tie it into what we just talked about up above, and then high transcription. Later on, we'll talk about how we measure transcription and how we measure acetylation. And so during the first 24 hours, there's no hypersensitive sites. None. During day 5, U, which is embryonic. So just very simply, where there's a DNase hypersensitive site, that should correlate with high gene expression. 14 days, the alpha D and the alpha A, those are two different kinds of hemoglobin, but they're both adult hemoglobin. And when we say they're different, they're just very similar, but slightly different sequence. In the brain cells, none are expressed there. Would you expect more or less acetylation in the regions sensitive to digestion by DNase? More, because acetylation leads to decondensed chromatin, and DNA decondensed chromatin is more sensitive to DNase. Alright, so we'll really make that one short, 12 minutes and 37 seconds, and we'll stop